Dr. Rooney, would you like to introduce our next speaker?
Yes, I can do that, Scott. Our next speaker is Ammani Kyanam. Ammani is a native of India. She made her way to Texas to do a master's degree back a few years ago. I don't exactly know when she started that, but she was working with David Stelly at the time. When she finished up with Stelly in the master's degree, she wandered over here, and we figured out that she wanted to work with us, and so we gave her a shot at it, and have enjoyed having her in the program. She's had a big week this week. She passed her oral prelims yesterday and given her seminar today, so I'm anxious for her to finish this up, do a good job, and then start writing some papers. So with that, Ammani, no pressure. It's all yours.
It's not on. Okay, sorry. Thank you, Dr. Rooney, for the introduction. Thank you, everybody, for joining us today at the end for the last session of seminar and the opportunity to share my current research. So today I will be talking about the evaluation of the effects of TFMSA on sorghum inbreds and their derived hybrids. So to give you a brief introduction, to generate diversity to select from, most breeders make crosses. But most crops are not dioecious, where the flowers, the staminate flowers are male flowers and pistillate flowers or the female flowers are born separately, and crossing these is extremely easy. However, cultivated crops don't usually always have this facility, especially in crops like cereals and sorghum, where the stigmas and the anthers are within the same flower, and these flowers are sometimes very small, making logistical issues when you're trying to emasculate. However, these crops are in fact sold as hybrids. And how is that done? Economical hybrid seed production is facilitated by the use of cytoplasmic genetic male sterility systems, where they use a combination of abnormal cytoplasm in combination with male sterility genes, nuclear genes, that results in a male sterile progeny that can be crossed with a restorer line that restores fertility so that you get a fertile hybrid. However, making a AB line pair that can sustain itself, which are isocytoplasmic, takes up to 5 generations of additional back crossing to get there, and we can't do hybrid testing until there is a male sterile pair. Additionally, there are genetic male sterilities available to make some of these crosses, but it's not always feasible. In the program, breeding crosses are usually made using a system called pour crossing, where the panicle is trimmed back and covered with a plastic bag to increase the humidity within around the panicle that prevents the anthers from dehiscing pollen, and then quickly crossed with the pollen source. However, this results in a line that's usually contaminated with selves. So therefore, the purity of the cross is not great. Additionally, having a gametocide, you can use it for something called early generation hybrid evaluations, where you can screen newly developed B lines early on to confirm their general combining ability to produce good yielding hybrids before you go through the back crossing efforts to develop AB line pairs. Additionally, a gametocide can be used for interspecific hybridization and doubled haploid production also in sorghum. So what is TFMSA? Expanded to be called Trifluoromethylsulfonamide, or TFMSA, as it is known in our program, was discovered by Dan Lussert at DuPont Pioneer. The mode of action of TFMSA is, while it was not confirmed, it is suspected to be with the interference of proline transport to the developing anther, which starves it of the essential amino acid, thereby resulting in male sterility. My colleague here, Nick Borman, he did a study where it was shown that TFMSA can be applied in the field and we are able to make crosses, and the effective dosage was anywhere between 10 to 15 milligrams per plant. So this gave rise to my study, where my research questions are to find out if this treatment of a line with TFMSA results in any fitness costs to the productivity of the treated line, and to evaluate if this derived hybrid seed quality and performance is comparable to the available control methods, which is the CMS system, and to see if this can be used for early generation hybrid evaluation within the program. So getting into the first study, which is the Inbred Evaluation, we used a random set of 23 inbred line pairs that were white-seeded. The reason we selected white-seeded was because we used pollinator lines that are red-seeded that you can see here in this picture. And the reason for this is because red-seededness is a dominant trait, and the hybrid can be identified by a phenotypic marker where, when crossed with a white-seeded female, the hybrid plots should all be red-seeded. So within the hybrid evaluation, we had 3 treatment levels: the A line, which was the control for the sterility system. It was cytoplasmically male sterile, and the G line, which is just a B line that is isocytoplasmic to A lines, but treated with TFMSA, and it shall be referred to as G line in this presentation to indicate it's any line treated with a gametocide. So the 3 treatment levels are A line, G line, and B lines. So coming to the dosage and application, the application was done using a backpack sprayer with a constant pressure of 15 pounds per square inch. The carrier solution included an emulsifier, methylated soybean oil, and a surfactant R11. And the application was before flag leaf initiation. So phenotypically, the sorghum plant here is still vegetative. And the reason we select prior to flag leaf initiation is so that we can apply the chemical prior to the plant turning into a reproductive stage, which is indicated by the flag leaf initiation. And the reason for application prior to flag leaf initiation is so that the complete panicle, remember sorghum is an inflorescence, so there are a large number of florets within it. So the reason for application of flag leaf initiation is so that the complete panicle will remain sterile. So the dose of 25 milligrams was split into 2 applications. The first application was done around 55 days after planting at the rate of 15 milligrams per plant. And the second application was done about 8 days after the first application at 10 milligrams per plant. So coming to the results. So the expression of male sterility. Here you can see 3 different panicles, one from a specific genotype laid out on a black background for 3 different treatments. If you notice, the B line here is the only line that has shed pollen. Oh, I'm sorry. Each of these panicles were tapped the same number of times on the table to show pollen shed. And as you can notice that the B line is the only line that is shedding pollen that is visible. So now if you zoom into the pictures and look at the anthers themselves, the B line anthers are bright yellow and usually plump, indicating fertility. When you compare it with the same genotype but treated with TFMSA, you see that the anthers are slightly shriveled, as indicated by the pointed end that you see here. And phenotypically, they are similar to the corresponding A line. So coming to the effects on the phenotypic effects on the treated line, we observed that across the lines, treatment with TFMSA resulted in a slight delay in maturity. Here I have a picture showing the same genotype planted. One of it was treated with TFMSA, the other was not. As you can see here, the untreated version shows that the flowers are, the inflorescence are heading, and that's why they're bagged and they are at the stage of tip flowering, whereas its corresponding line is just about starting to show the panicles. indicating this delay in flowering. Additionally, we also saw that treatment resulted in a reduction in height. So when compared with the corresponding A line, we saw about an average 16 centimeters reduced heights and about 12 centimeters reduction in when compared to the B line. So when comparing other yield parameters, we observed that the panicle length was reduced significantly by about a few centimeters, and we saw the same trend with the panicle yield and the seed set rates. So in coming to the seed yields, we noticed that the G line yields were slightly lower than the A line or the B line. And here I also compared the open pollinated types and controlled pollinations where bags were applied to the panicles. This is important because when we make breeding crosses, the panicle is usually always covered with a pollination bag. This results in a microclimate inside that can affect seed set. So that's why we compared both open pollinated and controlled crosses, and we noticed similar trends in both, where the G line had lower seed set. But even though this seed set is low, there is still 15 grams of seed per panicle that is produced using a G line or TFMSA treatment. These 15 grams of seed in sorghum can go a long way. 15 grams of seed, you can plant 5 different plots. So that's why this method is still very applicable as a breeding tool. So getting to the next step in my experiment, it's the hybrid evaluation. So of the 23 inbred lines that were planted, we were able to generate hybrids from 17 of them due to nicking issues with both the pollinators that were used. We used 2 different pollinators. They are slightly phenotypically different, and they are known experimental lines that are used for testing general combining ability of inbred lines within the program, and they were red seeded. That's why they were selected. So with these 17 inbred lines, we produced 34 hybrids that were planted in a replicated complete block design with 2 treatments: the A line, where the cytoplasmic male sterility was used to generate the hybrid seed, and the G line, where TFMSA was used to generate the hybrid seed. These experiments were planted in 3 locations in Texas, which are the normal locations where hybrid yield evaluation takes place: Monte Alto and Taft and College Station. So for evaluating seed quality of the hybrids, I did some germination tests using a ragdoll test. I selected randomly 10 hybrids, and I tested about 100 seeds in 3 replications per treatment, 3 treatments: A line, G line, and B line. Overall, the seed quality, the germination rates were around 70% and satisfactory, over 70% and satisfactory. There were 2 lines that had statistical differences, but the difference that you observe here is only for the B line, which is irrelevant for this study. And the one that we saw where the germination rates were very low, we observed that for all of our seed sources as well. However, this did not reflect in the stands that we saw in the field. The lines developed with the low performing germination rates had good stands in the field. So coming to evaluating the levels of selfing observed in the field, there were only 3 genotypes that showed selfing in both A line and G line treatments. They were used to compare the rates of selfing in both these sterility systems. The G line did not statistically differ in the number of cells that produced, indicating that the quality of seed produced using TFMSA is very good and pure. Comparing the phenotypic aspects of the hybrids, we noticed statistical analysis was done to compare all the phenotypic traits. None of them had any except for plant height. There were no differences between the 2 treatments, And when you look at the plant height, the difference, the numerical difference, even though it is statistically significant, it's still very small. And the most important trait that is relevant for hybrid performance is the grain yield, and it's not different for both the treatments, indicating that this method can be definitely used for hybrid evaluation studies. This is just a pictographic representation of the same hybrid that's produced using either the cytoplasmic male sterility or using TFMSA. We observed that the days to flowering and the plant heights of these hybrids are similar. So the next step is to identify if the inbreds are classified similarly with both the systems. So we noticed that the G line accurately classified the best performing lines, and the numbers were comparable even for the low performing lines, indicating that we can use TFMSA to evaluate if the inbred line produces acceptable hybrids, and select the lines that we would want to include for sterilization. So in conclusion, it's important, TFMSA can induce 100% male sterility when applied at the correct stage and at the correct dose. This is just an example where I'm showing a panicle that has a branch where there are sterile anthers in the first part, in the top part of the inflorescence, and there are fertile anthers in the bottom. So sorghum panicle flowers sequentially, and it takes sequentially from the top to the bottom, and it takes about 5 to 7 days to finish flowering. So what this tells me here is that this part of the panicle flowered the day before I took this photograph, and this part of the panicle is flowering today. And at this point, the reversion happened, indicating that the application, the correct dosage of TFMSA and the correct time to apply is critical in order to get complete sterility. So how do I know these anthers are sterile? When you look closely, you see that it's completely shriveled. If this anther were fertile, it would be a much darker reddish brown color. So answering my research questions, does TFMSA has an effect on treated inbreds? Treated inbreds do show some effects of treatment, such as reduced panicle length and statistically different average seed weights per panicle. But it's important to note that it is still producing a hybrid seed or crossed seed that we can use in sufficient quantities and great quality for the breeder to test. Does TFMSA has an effect on derived hybrid seed quality and performance? It does not. Hybrids produced using the G lines were phenotypically similar to those of the A lines, and there are no statistical differences in the hybrid yield performance. Can it be used for early generation hybrid evaluation? Most certainly. So how is having a gametocide in a sorghum breeding program valuable while there are alternative methods of emasculation already in place? So double haploids are technology that is evolving, and sorghum does have a few inducers. So in order to induce haploid generation, a sorghum line has to be crossed to an inducer. However, if we use the available methods of sterility, such as A lines, the resultant double haploid would be male sterile and would not be able to maintain itself, so you could not produce seed. The same is the case for genetic male sterility. It would not be able to maintain itself. So you could say, oh, there's hand emasculation, you could still be able to produce fertile progeny. However, the induction rate, the number of emasculations that can be made on a sorghum panicle average between 10 to 15 florets per panicle. And the induction rate is only 1 to 2% in sorghum. So in order to be able to get doubled haploids and the numbers we want, you will have to do a lot of hand emasculations, which makes it a logistical difficulty. So therefore, having TFMSA is an added breeding tool for a breeder. So this comes to the end of my presentation. Before I conclude, I would like to thank my committee, which includes Dr. Rooney, Dr. Trish Klein, Michael Thompson, and Dr. Kearns from entomology. Dr. Hodnett is not on my committee, but he has been instrumental in completing this project. He has assisted me and guided me throughout this project, and he's been out there with me spraying every time I had to. The sorghum lab, most especially because I had to phenotype, I don't even want to remember the number, over 2,000 panicles. And without these people, I could have never finished it myself. Thank you to my friends for giving me emotional support during my program, and all the funding sources, the AgriLife Research and Soil and Crop Sciences, and the Borlaug Scholars Program, and the Association of Former Students from which I received scholarships. So I finished the presentation, and if there are any questions, I am ready to take them.
Good job, Ammani. Very interesting research. Questions?
How do I see?
Dr. Ibrahim. You're muted. There you go.
Very good job, Ammani. Thank you. So the E line is basically a treated B line, right?
Yes.
And so when you compare the hybrids coming out of that, you compare the A line hybrids
Yes.
to the B line hybrids.
Yes, sir.
Why don't you compare the B line hybrids to the E line hybrids?
So I'm sorry, your question was, why didn't you compare B line hybrids with the G line hybrids?
Yes, yes.
So, sir, the B line is fertile. So the seed that came out of that is self-pollination. It is not a hybrid. So that's why we cannot compare.
Yeah, that's right. That's right. Sorry. I see that. That's true. Because the B line is a maintainer, so it produces.
Yes, sir.
So it's not a valid comparison. Yeah,
that's why we couldn't compare. So that's why we used the A lines, and mostly because A lines are what are used to develop hybrid seed.
Yeah, that's right.
So that is the standard in the industry. So that's why we use that.
And is this TFMSA? How costly is it? You know, can you use it commercially to produce hybrid seeds?
So it is. So TFMSA right now is very expensive in terms of for production of hybrid seed. So in terms of cost, I think it costed us about $1.50 to $2 per plot for just the chemical. However, if you were a company that is interested in using this, you could hire a chemist to produce TFMSA in bulk for much cheaper. However, sorghum is a tillering plant, and as I have mentioned, this induced sterility does break, and because of that, it causes issues with the purity of the hybrid seed, which is why I don't think supersedes cytoplasmic male sterility in production of hybrids. Do I make sense?
Thank you.
Thank you.
Dr. Rastani.
Hey, Ammani, very nice talk. The question I have is, what about other species? How efficient is this chemical with other species? And is there a difference between monocots and dicots in terms of male sterility?
So I know for a fact that it works in corn because that's where it was tested. I know Dr. Hodnett has tried it in other species, and I remember a conversation with Dr. Stelly where he told me that it did not work as well in cotton, and at that time we supposed it's because in dicots the transition between the vegetative phase to the reproductive phase is not as complete as it is in monocots.
Yes.
That is probably why it does not work as well. So I don't know if there are other crops that they have worked, that Dr. Hodnett has worked in. But I think for monocots, it might work. But for dicots, I don't know.
Okay, thanks.
Thank you.
Dr. Stelly?
Yeah, well, it's a good time to follow up. Yeah, we did not have any success with it in cotton, unfortunately.
Yeah.
Yeah, so I think you've summed it up well. I was wondering, well, thank you very much for your talk. It was nicely done,
you.
Ammani. I was wondering if you could go back to, I think it's your last slide before you got to your conclusions where you're contrasting the hybrids. Yeah, that one.
This one? Yes.
Yeah. No, I think it was one more. Were you looking, you had some ranks.
Oh, yes. Okay, I see the ranking.
Yeah, that one.
This one.
There you go. Yeah. So I was kind of curious, did you create some sort of a plot of these ranks, you know, putting one along one axis and the other along the other axis, just to graphically depict how they correlate with each other?
Actually, I did not, but that's a good idea to do. So
I mean,
yeah, I think that would be visually more appealing. I think I will try that. So I just ranked them based on yield. This was not like a statistical test, but the yield of the hybrids that the inbred line produced and how they compared between the two treatments.
So was there a reason you avoided using a statistical test of some sort?
So I only have 2 hybrids. So this is a small data set, and that means I only have 2 ranks to compare between. So that's why I didn't do a statistical test.
Mm. But you had what, 17 pairs?
Yes, I had 17 pairs. So what I mean is from each inbred line, I had only 2 hybrids.
Right.
So yeah, so that's why I didn't do a statistical test.
But well, anyway, I think you might want to look at that a little bit more closely. And for sure, the graph, I think would.
Yeah, I think that's a great idea. I will definitely look into that.
Yeah, and while you're at it, look, you may want to talk to a statistician to see if they can suggest something that would be legitimate and provide some statistical power to back it up even more. So.
Yes, I will do that.
Yeah, it looks very interesting. So,
thank you. I will do that.
Other questions?
Ammani, this is
Yes.
BB Singh.
Hello, Dr. Singh.
Very good presentations and good work.
Thank you.
You mentioned that you have to have 2 applications of the gametocid, 15 milligram per plant first and then 10 milligram the follow-up.
Yes.
So how do we ensure that the whole plot, big plotted commercial, will get this dose uniformly? Otherwise, there will not be 100% sterility. So how do you ensure that?
So, Dr. Singh, my personal recommendation is just to use TFMSA as a breeding tool. And the reason we split the dose into two application rates is because in a breeding nursery, there are different genotypes that mature at different times. So the reason we split the dose is that the chemical is there within the plant throughout the flowering season of the nursery that we have. So the chemical by itself is effective at really small doses. So the initial study that they did, it was effective even at the rates of 2 to 6 milligrams per plant. So the dose that I apply, 25 milligrams, is actually a lot higher than the plant actually needs. So the dose that we are applying, so even if your plant out of errors in application, it gets a little less than 25. If it's applied at the correct time, it will be sterile. Did I answer your question?
Yeah, very good. And what is the ratio of your A line versus G line? Number of rows alternate, and it is all air pollinated, or you make any
So,
effort?
so I actually handmade the crosses that I did in both the sets, because the size of the plots that we evaluated were very small, Dr. Singh. So they were about 20 feet long. So they had only 60 plants per plot on average. So I made over, I should say, over 20 crosses per plot. So I actually handmade all the crosses. So I carried pollen in a pollen bag.
Very good, thank you.
Okay, thank you.
Ammani, can I ask another question? What's the percentage of selves when you apply the chemical to the bee lines to create the
So
the year lines?
So the number of selves is less than 1% in the hybrid plots that
1%?
we observe. Yes, less than 1%.
Okay. And why did you choose a pre-flag leaf stage? I mean, can it be applied earlier than that?
So the reason we chose prior to flag leaf initiation is because Dr. Hodnett has done some research in this, and he found the optimum time of application to be prior to flag leaf initiation. So there's a paper from him in 2018 that talks about how he ended up there. But just to summarize it, it's just if he applied after the flag leaf emerges and the panicle is in the boot, he noticed that the top half of the inflorescence was fertile, whereas the bottom half was sterile. So there's no complete, like the whole panicle is not sterile. So based on all of these experiments, he found out that application prior to flag leaf emergence is optimal to get the full panicle sterile.
Thank you.
This is Dirk. I have a quick question.
Yes,
Is
Dr.
I may have missed it, TFMSA, is it a salicylic acid?
No, I don't think so. It is a trihalogenated I mean I had it in my other presentation, but it's a trihalogenated amide. So in this, I don't have a picture of the chemical by itself, but I don't think it's an acid.
It's, I mean, a salicylic acid,
Yeah,
which is
yeah.
a plant hormone. It's not a salicylic acid.
No, I don't think so.
Okay.
Other
If you have
questions?
that chemical structure, you could send it to me. It'd be great.
I will email it to you.
Thanks.
Other questions?
Monty,
Okay.
this is David. I'm just curious. You talked about that it works in a breeding program, and just wondered, when you apply it to something like Dr. Rooney's program where he's making X thousand hybrids a year, how many man hours would you project it save? How wonderful is it?
So as of now, I actually don't have math on how much time it saves us, but I can tell you it has increased the size of our selection nursery by a lot, because we worked an extra month this year. So I actually don't know how much time it will save in reducing the number of sterilization crosses that we are making, but I think we will compensate it by increasing the amount of time we are selecting more new lines. So I don't know, maybe Dr. Rooney can jump in and answer.
He's not jumping.
So I'm on my own here. I don't know, I think.
That's a fair answer.
Yeah.
I just think it's worth thinking about how much.
I'm here. I'm here. It does several things, David. First of all, there's crosses that we physically cannot make without it. So we can move things faster than we could in the traditional sense that we did that. So you have the savings of time, and it reduces the amount of things that we carry forward in a sterilization process. And as Ammani mentioned, if and when we actually start integrating double haploids, which I think probably will happen, Without TFMSA, you cannot do doubled haploids. You had to have a gametocide to do that. So most of it is about functionality more than, and there is certainly time savings and number savings, but some of it is just sheer functionality.
Thank you.
Sure.
And
All right. Well, sadly, this brings us to the end of our seminar series.